In this episode, we learn about the T cell's past. The T in T cell stands for thymus, the location where T cells mature. The thymus comprises multiple capsules, each which consists of the inner medulla and the outer cortex. After hematopoiesis, immature T cells, known as thymocytes, move from the bone marrow to the thymus to mature. Thymocytes start off at the cortex as double negatives, expressing neither CD4 nor CD8. Here, the receptors are created. T cell receptors consist of two chains, the alpha chain and the beta chain. The alpha chain consists of variable and joining subunits, while the beta chain consists of variable, diversity, and joining subunits. Each subunit is encoded by multiple genes. The alpha chain has 42 V fragments and 6 J fragments, while the beta chain has 48 V fragments, 2 D fragments, one which corresponds to 6 J fragments, and the other corresponds to 7 J fragments. However, only one gene fragment is needed for each subunit. During receptor formation, genetic recombination occurs. The D fragment joins to the J fragment, while the V fragment joins to the D or J fragment. This creates a T cell that produces only one receptor for the rest of its life. The gene fragments give 252 possible alpha chain configurations and 624 possible beta chain configurations, for a total of 157,248 possible T cell receptors. However, the joining process is imprecise. Some additional nucleotides may exist between these segments. Some nucleotides of the gene segment may be accidentally spliced. This further increases the number of possible T cell receptors produced. Thymocytes subsequently express both CD4 and CD8, becoming double positives. They go through a process of positive selection, which tests their affinity to cell surface receptors MHC on macrophages and dendritic cells. Binding to MHC results in survival signaling. Thymocytes that fail to or weakly interact with MHC would thus die. Thymocytes that interact with MHC1 will become CD8 positive T cells, killer T cells, while thymocytes that interact with MHC2 will become CD4 positive T cells, helper T cells. Single positive thymocytes move to the medulla, where they undergo negative selection. Due to the random nature of genetic recombination, it is very likely that autoreactive T cells which target self-antigens are produced. Thymocytes are screened for their interaction with self-antigens by thymic epithelial cells through MHC1 and antigen-presenting cells through MHC2. Those that interact with self-antigens are removed. This maturation process is known as central tolerance, which removes lymphocytes that target the self. This prevents a situation known as autoimmunity, where the immune system mistakes your body as a threat and mounts an immune response against it. The selection process, however, is portrayed in reverse in this episode. The remaining thymocytes enter circulation as naive T cells. The maturation process also generates regulatory T cells, which control T cell activation after prolonged periods of immune response. Regulatory T cells also inactivate any autoreactive T cells that survive the negative selection process.